Hello everyone, this is Channel 2, the AGU Plenary Lecture. I'm Masumi Zaiki, Seikei University, moderating this lecture. Today we have Professor Hartnett from Arizona State University. Her presentation today entitled Exoplanetary Ecosystems. What do we know and what do we need to know? So, Professor Hartnett, would you please begin your talk?
Thank you very much, Masumi, and thank you very much to the organizers. I'm so pleased to be presenting today at the JpGU-AGU joint meeting. And my talk about exoplanetary ecosystems will be a bit of a mix of biogeochemistry, which is my primary field of study, and I collaborate with astrophysicists and geophysicists. And I think we've begun to develop a really novel new way of thinking about how we're going to search for life on exoplanets. Before I forget, I do need to thank my collaborator, Natalie Hinkle, who's an astrophysicist, and you will see her work in the stellar abundance data that I'm showing. Several research groups from the NASA NExSS team, the Frontiers in Earth Systems Dynamics team, and the Hydrothermal Organic Geochemistry group at ASU, and my own research group, the Carbon and Nitrogen Dynamics Lab, or CANDY Lab, this is our logo. I can't do anything without my students. They really are the primary reason why I do science, and they've been tremendous contributors to this work. So today, my talk is in 3 parts. I'm going to talk to you about elements in stars and planets, and I think by the time I'm done, we'll realize that this is a highly data-limited problem. We really do need more data on elemental abundances. I'm going to talk about exoplanetary ecology as a way of thinking about the emergent properties of living systems. And I'm going to close with an example from a water world, a planet with much more water than Earth, and talk about the detectability index for oxygen as a biosignature. So hopefully, we'll see a thread that goes from how we understand the composition of stars and planets to the types of processes that happen on planets, and an example of a type of planet that we don't have in our own solar system, but for which we can write models for what biosignatures might look like. So here we go. First takeaway message for this talk, if you remember nothing else, we know almost nothing about phosphorus in stars or in planets, and phosphorus is critically important for biology. Other elements are important too, and I'll mention a few of them, but primarily I'm going to talk about phosphorus today. Second take-home message, if we can detect water on an exoplanet, it's probably too much water. And that is counterintuitive. We tend to think that if planets have water, that makes them habitable and we should search for life there. But the amount of water does matter. So elements in stars and planets. When we look for life on other planets, we traditionally look at the habitable zone. The habitable zone is a region around a star where there can be liquid water at the surface of a planet. And so this is our own solar system. Earth, where we live, is firmly within the habitable zone. Venus is right on the inside edge and probably too warm. And Mars is currently at the outside edge of the habitable zone, and it's generally too cold. But detecting life will be more complicated than just knowing if a planet is in the habitable zone. The habitable zone is not the whole picture. Planet composition matters a lot, right? What elements are present on the planet will make a very big difference to whether that planet is habitable. Life detection, more so than just the composition of the planet, is going to depend on the rates of the processes that biology conducts. So essentially planet-scale metabolism. This is not just is a planet at the right distance from its sun. We can make some guesses about habitability for planets, right? Rocky planets like our own are sometimes habitable. Earth certainly is, but Venus may not be. Gas giants, also an example of a planet in our own solar system, are probably not habitable. Icy bodies like Europa or Enceladus, perhaps, maybe are habitable because they have a liquid water ocean and potentially geothermal heating. But for exoplanets, planets around other stars, we really don't know. How are we going to decide if they're habitable? And this is particularly challenging for planets that we don't have examples of in our own solar system. For those planets around other stars, we will have to assume that the planet's composition is a lot like the composition of their stars. Fortunately, we have a very good database for stellar composition. The Hypatia catalog is the largest catalog of elemental abundances for nearby stars. And by nearby, I mean within about 150 parsecs of the sun. This is our sort of near neighbors in the galaxy. The Hypatia catalog has measurements of elemental composition for about 6,200 stars. This is a bar chart that has the number of stars with a measured element plotted for each of the elements that have been measured. And about 73 elements have been measured in stars, and each star has iron and hydrogen plus at least one other element. So this is quite a nice data set. There are examples of rock-forming elements like magnesium, silicon, calcium. And you can see here that these elements are represented in 5,200, 5,600, 5,400 stars. There's a lot of data for these. The bioessential elements that I'm more interested in, carbon, nitrogen, phosphorus, some have fairly large numbers of measurements. There's 4,500 carbon measurements, maybe 1,500 nitrogen measurements, but phosphorus here has only 92 stars that have phosphorus measurements at all. This suggests that if we're interested in phosphorus, we don't have a whole lot of phosphorus data to look to. So why are we interested in phosphorus? Well, from a biology standpoint, phosphorus is critically important. And life's composition, and there are many compositions for life, but we're going to generalize a little bit. Photosynthesis from algae in the ocean has a fairly fixed stoichiometry, a fairly fixed ratio of elements that are needed. So you need 106 moles of CO2, 16 moles of nitrate, 1 mole of phosphate, some water, some protons for charge balance, and you make this horrible looking molecule, which is just a representation of organic matter, but it has the same carbon, nitrogen, phosphorus ratio as the starting materials, and you make oxygen. In this case, phosphorus is a limiting reagent. So if the elements are not present at the correct molar ratio, the reaction can't happen. It's like baking a cake. If the cake recipe calls for 4 eggs and you only have 3 eggs, you can't make a cake, or you have to scale down that cake proportionately. So the elemental ratios, the molar ratios of carbon, nitrogen, and phosphorus in plankton are 106 carbons to 16 nitrogens to 1 phosphorus. And here's that equation at the bottom in case you want to remember it. We can do the same calculation for various bodies in the solar system. Earth's crust is relatively nitrogen poor, and so its carbon, nitrogen, phosphorus ratio is 5 to 0.05 to 1. So these are normalized to phosphorus. And that makes sense. Nitrogen is distributed in the solar system mainly in the gas giants, and it's fractionated during differentiation of the planet. I can do the same calculation for Mars. This is based on a recent published model of bulk silicate Mars composition. And you can see that Mars is actually carbon and nitrogen poor relative to the Earth. I didn't put the Earth's bulk composition into this table, but it looks somewhat like Mars. Our sun, on the other hand, has substantially more carbon and more nitrogen relative to phosphorus than plankton or the other planets. But interestingly, our sun doesn't necessarily look like all the other stars in the Hypatia catalog. The Hypatia catalog average star has a ratio of 800 to 280 to 1, and so it has less carbon, or the average Hypatia star has less carbon and less nitrogen than our sun. So if the relative ratios matter for the composition of life and for the composition of planets, we really want to know these things pretty well in the stars. So here's the data plotted as nitrogen-silicon ratio and carbon-silicon ratio. And there are 1,470 stars in the Hypatia catalog that I can do this calculation for. And you see that there's quite a general spread of data with a large group here in the middle. Our sun is way over here on the right-hand side of this plot. These are molar ratios. This is not the typical way that astronomers plot this. Usually astronomers plot these normalized to the sun, and so the sun's always in the middle. But by plotting them as molar ratios, we can see that our sun is slightly different from the average star in the catalog. Plankton plot about here, and the Earth and Mars plot over here very close to the axis, mainly because they have a lot of silicon and not much carbon and nitrogen. But I started out talking about phosphorus, so let's look at this plot with carbon, nitrogen, and phosphorus. Well, the first thing you see is there's only 50 stars for which I can do this calculation. Only 51 stars have both phosphorus and nitrogen data. Now it is extremely difficult to say what a general pattern is, right? There's a little bit of a group of data here, and our sun appears to be roughly similar on a sort of carbon to nitrogen ratio basis, but substantially higher in a carbon to phosphorus ratio, which implies that the phosphorus is quite low. Earth is over here, Mars is here, and plankton is down here. And the reason there's so few stars with phosphorus is very difficult to measure. It has weak spectral lines, there's substantial interference from water, and you need a high resolution spectrograph with a lot of telescope time. So at the moment, we really don't know much about phosphorus composition in stars, and so we'll have a very hard time making predictions about phosphorus in planets. So let's summarize this section. Molar ratios determine the chemical reactions. It's important for rocks, and it's important for biology. It was difficult to put the stellar abundances on a molar basis because you have to undo a variety of normalizations, but it can be done. And we need to predict ecosystem dynamics for planets based on these stellar compositions, but we're missing key elements. In particular, we're missing phosphorus and nitrogen, and we really do need more phosphorus data. So exoplanetary ecology. The lack of data shouldn't stop us from thinking about exoplanets. We can look to the Earth and we can build models. And ecology is actually has been a key part of thinking about Earth system science for a long time. As early as Darwin and his voyage on the Beagle, he is reputed to have taken Charles Lyell's Principles of Geology with him on that ship. And you can see in his notebooks beautiful maps, and he had a clear understanding of how organisms evolve in their environmental context. This is critically important for thinking about planets, particularly at large scale. Most of us can't think at a planetary scale very well. So I'm going to use this example of a Winogradsky column as a sort of a small scale version. If you imagine this had a top, you can have algae growing in the surface water, you can have bacteria in these sediments that use oxygen, further down you have bacteria that use nitrogen, and further down you have bacteria that use sulfur. So there are many metabolisms in this one system. And if you measure the amount of oxygen in the headspace of this column, it integrates all the metabolisms that produce oxygen and consume oxygen, as well as any abiological or non-biological processes that might be consuming oxygen. So we can imagine that the atmosphere is like the headspace of this Winogradsky column, and we need to infer all of the processes that change the atmospheric composition. Those processes could be biology, but they could be lots of geochemical and geological processes. Degassing from volcanoes, serpentinization, which is a water-rock reaction that produces hydrogen, surface weathering of sulfide minerals or other kinds of minerals that become oxidized, and again, biological production. So all of these are affecting the oxygen content of the atmosphere. We will need models that integrate all of these geological processes with biological processes, and ideally, the processes that I just told you about are really at the surface. They have to be integrated with models of mantle dynamics and atmospheric chemistry in order to really have an understanding of how planets work. Because for exoplanets, we're just going to have a glimpse of their atmosphere, and we need to be able to understand how all these processes are happening. At this really large scale, we won't see the ecosystem. We won't see trees. We won't see a protein or a microbe. What we may see is the influence of a microbe or lots of microbes, but only if the biological processes are happening at a rate that's fast enough to give you a signal larger than the geochemical rate. And so you can imagine, in a net sense, the planetary biological production is the net production of, say, oxygen by biology, minus any net production of oxygen by geochemistry. So we really do need to understand both biological processes and geochemical and geophysical processes. So to get at a way to quantify these two, the biological term and the non-biological term, our team has developed what we call the Detectability Index. When you think about a biosignature, for example, oxygen gas in the atmosphere, whether a gas is biosignature comes not only from the probability that life produced it, but also from the improbability that a non-biological process produced it. In other words, you not only need to know that it was made by life, you have to be sure that it's not very likely for it to be made by a geological process. And this is a quote from Mustard. I use it a lot. So the Detectability Index on a log scale, so that you can look at large variations in numbers. We have calculated as the net biological source of oxygen, so that's photosynthesis and respiration, divided by the sum of any non-biological oxygen sources. And a non-biological oxygen source in particular is photolysis, right? UV light breaks down water and creates hydrogen and oxygen. The hydrogen escapes to the atmosphere and the oxygen is left behind. So now we have a tool to think about this planetary scale ecosystem. Let's apply this tool to a water world, a planet with a lot more water than the Earth. These types of planets, the water worlds, are not science fiction. We know they exist. This is an artist's rendition of the TRAPPIST-1 system. And the TRAPPIST-1 system has several planets. Several of them are potentially Earth-sized. Some of them are in a habitable zone. What's interesting about the TRAPPIST system planets is that they have a lot of water. Current best estimates of their water contents are somewhere in the neighborhood of 5 to 15% by mass, so weight percent. Some of them have much higher masses of water, maybe as much as 50%. And the latest estimates for the drier ones is that they may have less than 1% water. Those estimates change as we get better estimates of the planet mass, and they depend on assumptions about the greenhouse state of those planets. But the TRAPPIST planets could range from, let's say, greater than 15% water, which is extremely high, to a little less than 1% water. Our Earth, it has about 0.02% water by mass. We're actually quite dry, even though we have a liquid surface ocean. So some of the TRAPPIST planets might be potential examples of water worlds where the planet has liquid water, it's warm enough because it's near to its sun. We can look at whether or not such a planet is habitable, or if life on such a planet would be detectable. So we're reasonably certain that lots of planets are going to have reasonable amounts of water. This is a plot by Raymond et al., and it's a model that shows numbers of planets on the y-axis, and there are planets with less than 1 Earth's ocean of water. These are dry. They call them Mars-like. Planets with 1 to 10 Earth oceans of water. So these are Earth-like, and we know these are habitable. Water-rich planets that have 11 to 100 Earth oceans, and true water worlds with more than 100 Earth oceans of water. If these planets are close enough to their stars that they're warm, and if these planets have all of the requisite elements for biology, they could be habitable. We can apply our detectability index to a water world planet and ask, what's the likelihood that oxygen is a good biosignature? We'll do this as a thought experiment. So we'll start with the Earth. This cartoon is a model of the Earth's biogeochemical cycles for oxygen. And it's based on a paper by Catling and a recent paper by my graduate student, Donnie Glazer, that came out this year. And what you can see is that photosynthesis and respiration are big fat arrows. These are large fluxes, about 10 to the 4th teramoles of oxygen per year, being produced through photosynthesis and consumed through respiration. And there's a net export of 20 teramoles of oxygen to the atmosphere every year. So that's the biological production term. There's also abiotic production due to photolysis. And that photolysis term only produces about 0.02 teramoles per year. So the biological production term is much bigger. And you can calculate the detectability index for oxygen. The biological production is 20. The geochemical production is 0.2. And this calculation yields an index of +3. So it's a large positive number. And this makes sense for Earth. We know that the oxygen is overwhelmingly produced by biology. So imagine a water world where now the oceans are much deeper. They cover up the continents. For a planet the size of Earth, you only need about 5 oceans of water to submerge the continents. And when this happens, because the continents no longer have subaerial weathering, right? You don't get rain on the continents and rivers carving down mountains. The supply of nutrients and sediments will be much lower than it was on the Earth's ocean. This is important because phosphorus, the main limiting nutrient for photosynthesis in the oceans, is a rock-derived element. Predominantly, it comes from apatite. This picture is a nice gem quality apatite. It has beautiful blue color, but it's a phosphate mineral. It's a highly insoluble mineral. And when I say highly insoluble, the KSP, the solubility product for apatite, ranges from 10 to the minus 42 to 10 to the minus 116. Not very soluble. Now, surface complexation reactions help and increase the solubility, but still maybe no more than 10 to the minus 25. Because of this, on Earth, the phosphorus supply from weathering is pretty small, 0.07 teramoles per year. On our pelagic planet, where we've shut down the subaerial weathering, the continental weathering, we predict that the phosphorus supply is going to be about 2 to 3 orders of magnitude lower. That reduced phosphorus supply is going to have a big impact on the biological processes in this ocean. Less phosphorus means less photosynthesis. So now, the rate of photosynthesis and respiration is much smaller. The net export of oxygen to the atmosphere is now only 0.02, and that's about 3 or 4 orders of magnitude smaller than for our Earth. We can recalculate the detectability index for this water world. Now, let's just remember the Earth side. The biological production on our Earth is 20, and the photolysis production is 0.2, so we have a large positive detectability index. On the water world, you see my continent is submerged, sediment and nutrient supply is lower. The detectability index is about 0, right? The biological production of 0.2 is about the same as the photolysis production of about 0.02. Now, it's very difficult to tell if the biological production is bigger than the abiotic production. Oxygen is not such a good biosignature on this planet because we can't be sure that the oxygen we detect comes from biology. This is not exactly what you would expect for a planet that has an ocean, that has light, has some nutrients. But in this case, it's not a sure thing that oxygen is a biosignature, mainly because there's not enough phosphorus to maintain high rates of photosynthesis. More water than the 5 oceans that I added in the first part of the model doesn't make it any better. So this is a water world with 40 times Earth's ocean, getting closer to something like one of the TRAPPIST planets. In this case, the ocean is much deeper, deep enough that you form what's called a high pressure ice mantle at the bottom of this ocean. That high pressure ice mantle really reduces weathering to almost nothing, particularly on these continents, and it may even reduce water-rock interaction at the seafloor. In this case, we don't have a good estimate for what the photosynthesis and respiration rates might be, but they're even lower than they were for the 5 oceans planet. So now let's just say that it's 0.002 biological production and 0.02 photolysis production. Well, now the abiotic production is larger. The detectability index is a negative number. And if this number is actually 0, it will be an infinitely negative number. On a planet with 40 times Earth's oceans, oxygen is a terrible biosignature. It's only a signal of photolysis. So this 40 times Earth's ocean corresponds for a planet of an Earth-sized planet, about 1 weight% water. It's not really all that much water. So what have we learned? I've gone through a bunch of calculations. The detectability index can help us prioritize planets for which oxygen might be a good biosignature, right? Oxygen is one of the best biosignature gases we have because we know it's produced by photosynthesis, a biological process. We can compare the detectability index on Earth with one Earth ocean. We calculate it based on modern Earth to be 3, and a big positive number is a good biosignature, so there's pretty high utility. On our pelagic planet, our 5 oceans of water planet, the detectability index was lower because oxygen production was lower. Remember, that planet doesn't have as much phosphorus supply, and so it can't do photosynthesis at the same rate. Its detectability index was 0 or 1, and that means the biosignature is only fair. We won't be certain that oxygen is produced by life. It could be produced by an abiotic process. And on the water world with 40 times oceans, the detectability index is less than 1. Oxygen's a really poor biosignature. So now we have a way of quantifying how well we may be able to determine whether a biosignature we measure in the atmosphere is due to biology or not. This is really important because we need to be able to prioritize those planets for which we think the search for life is going to be fruitful. So now let's look at this model again. We know that Earth-like planets and these water-rich planets with liquid oceans could all be habitable, but it's not clear that life on those water world planets will be detectable because the production of oxygen will be lower. And it's lower not because photosynthesis can't happen. It's lower because photosynthesis will be limited by nutrient supply. This leads me to a cautionary point. Life needs a lot more than just phosphorus. We saw that in the photosynthesis equation. You can, in fact, write that equation for all kinds of elements that are important for life, right? Here's the carbon, nitrogen, phosphorus part of the stoichiometry. But plants and algae also needs sulfur. They need some of the rock-forming elements. So these are the bioessential elements, carbon, nitrogen, phosphorus, sulfur. They need small amounts of these rock-forming elements. It turns out they need even smaller amounts of redox-sensitive elements like iron and copper and molybdenum. And for modern ocean biogeochemistry, we spend a lot of time making sure that we can get the chemistry right. For exoplanets, we're in a much more difficult position, right? We're never going to know the detailed stoichiometry of trace metals or even some of the rock-forming elements dissolved in seawater of these oceans. And that means we're going to have to be thoughtful about how complex the models that we build are. So, in summary, we're going to have to estimate what we know about our exoplanets from their stellar compositions, right? We will not have good data for the exoplanets themselves for a very long time. And so we will need to predict phosphorus and nitrogen and these other bioessential elements from the stellar compositions. And we have almost no data for phosphorus and nitrogen in these stars. So the first thing we need to do is we need to build our catalog of elemental abundances for bioessential elements. The other thing I told you about today is that the types of planets that we may discover around other stars, we don't have examples of all of them in our own solar system, right? We know what rocky planets look like. We know what gas giants look like. But water worlds, which intuitively sound like they could be habitable, and they very well could be habitable, too much water may be a problem for life detection on these planets. Our group has determined a detectability index that can assess what types of planets oxygen will be a good biosignature. And we showed that phosphorus limitation on water worlds, planets with a lot more water than Earth, that phosphorus limitation can cause oxygen production to be much lower. So even though the planet is habitable and could be inhabited, there's not enough phosphorus, and therefore the rate of oxygen production will be small enough that you could be confused by the production of oxygen from an abiotic process like photolysis. So that ends my talk. I'd be happy to take questions, and thank you for your time today.
Professor Hartnett, thank you very much for your very interesting talk. So now we'd like to receive several questions or comments from the audience. Please indicate your intention in a chat window.
There's already a question there.
Okay, so Hilairy, can you check the question in a chat window?
Yes. Should I read the question? So the question is from Bill McDonough, and it says, thank you for your thoughtful presentation. Most, 90% of phosphorus is in the core, as it is a highly siderophile element. How important are magnetospheres? Thank you, Bill. It's a really important question. I totally agree that most of the phosphorus is in the core, and that's a huge problem from a life standpoint, right? The fact that there's just a thin skin of phosphorus at the surface, how we got it there is hard to ask. The importance of the magnetosphere, this is a little outside of my depth, but I think it's going to be, if it's important, right? Remember, we still only have one example of an inhabited planet. And that inhabited planet has a core. It has a differentiated core with a liquid and a solid inner core. Liquid outer core, solid inner core, and it has a magnetosphere. So if all of those things are prerequisites for a planet that can develop life, then it could be really important. I guess if my intuition is that core formation matters a lot to the distribution of elements. So it's entirely possible that the magnetosphere will matter as well. But I would ask my friend John Hernland for a really detailed answer to that question. Thank you for the question.
Thank you. Any other questions or comments? If you have comments, you can just, another question and comments coming.
So the next question is from Brooks Hansen. It says, on Earth, ocean hydrothermal systems remove phosphorus from the ocean. On a water world with different chemistry, could this reverse? That's a really great question. And we worried a lot about what to do with the hydrothermal phosphorus flux. In high temperature hydrothermal systems, phosphorus for sure is taken out of the seawater. In low temperature hydrothermal systems, it's not so clear if that flux is to the ocean or from the ocean. So the answer to the question is, on a water world with different chemistry, it could matter a lot if the relative proportion of high temperature sort of on-axis hydrothermal systems versus off-axis diffuse flow stuff. It could be really different. Yeah, and we don't have particularly great ways of estimating what the hydrothermal systems will be like on ocean planets yet. Oh, Bill comments that cosmic rays are really harmful. Yes, I guess I didn't take it all the way to that end point. If you don't have a way to protect your planet from cosmic rays, you will have a hard time building a biosphere, I think.
No? Okay. Again, thank you very much for your lecture. Everyone, thank you for coming today, joining this lecture today. And now we close the AGU plenary lecture. Thank you very much.